All right, everybody. Today's going to be a doozy of a lecture. We're covering several different phyla. So from the phylum Nematoda to the phylum Onychophora and the phylum Arthropoda, and then tons of different taxonomic groups within those phyla. So just hold on. It's going to be a lot of talking for a long time. So everything that we're talking about today is going to be in what we consider a superphylum called Ecthesis. And so everything in Ecthesis is going to be shedding or molting and shedding out of an exoskeleton that's made out of chitin as they grow. So as they're growing, this rigid exoskeleton becomes too small for their bodies, and so they have to shed it or molt out of it in a process called ecthesis. So if you look at this diagram of a crayfish or a lobster or whatever, I believe it's a crayfish, it's shedding out of its old carapace, and then it has this really soft new layer of chitin underneath that's still covering its body, and so it's getting rid of this old shed right here. So everything that we talk about today is going to be in this monophyletic group called Ecthesis. So if we look at our phylogenetic tree under this this class or the superphylum, we have two phyla listed. We have the phyla Nematoda and we have the phylum Arthropoda. So I've included another one right in the middle here called Onychophora. We do have a couple specimens for it. It's an extremely small group called velvet worms. So we'll jump right into the first phylum. So the first phylum is nematodes. These are roundworms, and so these are mostly free living, but there are some parasitic species. So out of the 25,000 or roughly 25,000 known species, only around 35 are actually parasitic to humans. But as I mentioned, there's about 25,000 known species, so that's a fairly diverse, extremely diverse group. But even this is probably a gross underestimation of the diversity here. So in a single teaspoon of soil, you can get thousands of nematodes. So the true number of species is likely over a million. So it's a hugely diverse group that no one's really taken the time to go through and describe many of the species. So they are ecdysozoan protostomes, so they go through the process of ecthesis. They molt that external cuticle. Let me see if I can pull up my laser pointer. So this cuticle out here is molted once they've grown out of that, or they start to grow larger in size, and that cuticle becomes restrictive. So they molt out of that cuticle and leave a endocuticle underneath, which is much softer, and then that becomes hardened over time, and the process repeats itself. They're also protostomes, so they during development, that blastula or that morula, the first opening to the digestive system is the mouth. They do have a complete digestive system, so they do have both a mouth and an anus. And they are dioecious, so this is one of the first groups that we'll talk about that has a fairly distinct male and female, and so they express what's considered sexual dimorphism. Males look nothing like females. You'll see this in the lab. So males will have a copulatory hook or copulatory spicules here at the end. Females will not. Females be much larger in size. Males be fairly small. So they're pseudocoelomates. They don't have a true body cavity. So if you look at the little diagram here, this water fluid, this fluid-filled area isn't considered a true coelom because it's not surrounded by mesoderm. It has both dorsal and ventral nerve cords, similar to the ligochetes that we dissected in the lab on Wednesday or Thursday last week. And so just like the oligochaetes, they have a hydrostatic skeleton. They're using the water or the fluid or the pressure of the fluid inside their bodies to maintain a sort of rigid body shape. Unlike the annelids, this phylum's not segmented, so you don't have these separations of the body. So if you ever see a difference between nematode and annelid, just know that these nematodes don't have segments. They're very smooth looking. So here is probably one of the most popular nematodes. So this is Canarhabatus elegans, or as many of you probably know it as C. elegans. And so this is a model organism for many research studies, especially genomic, because it's extremely easy to rear. So they don't require a whole lot of attention to grow them. It's also an extremely simple creature, so it only has 928 non-sex cells. And then the actual sex cells vary from, I believe it's like 30 to 100. So it's easy to measure the differences or changes that occur in the body of this organism. It's also easy to observe those changes. So it's see-through, it's transparent, so you can just look through a microscope and see what's going on within each cell or within organs or bodies. So this is probably the most famous or the most popular. It's free living. Many of the other parasites, parasitic species like Ecthesis lubricatus, which you'll see in lab, or actually you'll be dissecting in lab, it's parasitic to humans. I did make a mistake earlier in the semester. I was mentioning Trichinella, and so Trichinella is not a flatworm like I was talking about. It's actually a nematode, so that was my mistake, but it is still present in pork products, or was present in pork products in the US until it was eradicated, and that's a pretty nasty parasite to have as well. So this is the phylum that I've kind of slipped into here. So this is part of what we consider Pan-Arthropoda. So if you think about the phylogenetic tree that I just showed you, the one that you're used to seeing from your textbook or from the lab manuals, it has Arthropoda and Nematoda branching off from each other. But where we see that branching off between Nematode and Arthropoda, we can really consider Arthropoda as this group Pan-Arthropoda. So this group, Pan-Arthropoda, is including tardigrades, Onychophora, and the Arthropods. So it's a fairly diverse group that we don't have time to cover all the organisms, but I just wanted to slip this group in because we do have a specimen from, I believe, like the 1990s. So these are the velvet worms. It's a fairly non-diverse group, so roughly 200 species from 2 different families. So the reason why this group is fairly important is because it's the closest living, one of the closest living relatives to the arthropods. So they have a whole lot of similarities. They have a segmented body plan with a ton of appendages called onychopods, or stub feet. And so they're also similar in having a chitinous exoskeleton that they molt once they grow out of it. And they have similar respiratory organs, which we'll talk about later, and they have a very similar embryonic development. So one neat aspect of these organisms, or this phylum, is that they have a fairly complex social life. So these velvet worms will live in groups of 10 to 15 individuals that live and hunt together, and then once they take down a prey item, the lead or dominant female is the first one to eat. So we consider that a matriarchy within a social hierarchy. So they're a neat group of organisms, and there's a couple neat videos. If you're a fan of David Attenborough, he narrates one where they have a velvet worm hunting, and so they can exude the slime out of what looks like antennae, and they use it to trap prey, which they then eat. So this is a neat phylum. the next phylum, and by far the largest one that we'll talk about this entire semester in terms of just sheer diversity, is the phylum Arthropoda. And so Arthropoda simply means jointed foot. And so if you look at the legs of an arthropod, they'll be jointed. So there'll be tibia, you'll have tibia, femur, tarsi, and a couple other segments. So if you look at this grasshopper, you have several different joints, and even what appear to be like hands, those are jointed as well into claws, into tarsal segments. So everything in this group will have extremely jointed legs. So once again, we're ecthesis and protostomes. So every single organism in this phylum will be shedding that outer cuticle and molting through the process of ecthesis, and then it's also going to be a protostome. So the first opening to that morula or that blastula is going to be the mouth. So once again, we have segmentation. So this is important. The body plan of an arthropod is generally broken up into 3 different groups: the head, thorax, and abdomen. You'll see in 2 of the classes that we go through that is generally cephalothorax and abdomen, and then head, thorax, and abdomen is distinct for hexapods, which we'll talk about. So we have tagmosis, and so this is the fusion of segments. When we talk about Myriapoda or the centipedes or millipedes, they look just like an Onychophora or a velvet worm. But over time, we have these segments that are fusing together through Hox genes, which are genes, regulatory genes that are coding for certain regions that are condensing into certain regions, such as the head, thorax, or abdomen. So we consider that that condensation of these regulatory genes and the fusion of these segments as tagmosis. So once again, we still have a chitinous exoskeleton that's characteristic of Ecthesizoa. We consider that a cuticle, and so if you look at this cross section of a crayfish, you'll see this outer layer. I don't see it labeled, but this outer layer is all made out of alpha chitin or chitin. It acts as an exoskeleton, providing support for the body and muscle attachments as well. Already mentioned jointed appendages. So these guys have an open circulatory system. You'll see this in the crayfish, I guess Wednesday or Thursday. But let me pull up my laser pointer. We have a heart that's at the dorsal side of the body, and blood's flowing down to the ventral side through a vein, and then it gets oxygenated either through a book lung, trachea, or gills, and then from there it gets passed up to the heart, where it sits in this pool waiting to be sucked back into the heart and then transported throughout the rest of the body. So they're pumping this hemolymph, which is what we consider circulatory fluid for organisms with an open circulatory system. They're pumping that hemolymph from that opening in the heart down, and the cycle repeats itself. So we have distinct male and females. There's a few, well, there's always an exception in biology, but there's a few exceptions, such as with parasitic wasps and honeybees, or not honeybees, another group in Hymenoptera, so wasps. And then they're eucoelomate, so they do have a true body cavity around that mesoderm. So this is where taxonomy starts to get a bit confusing. So we're talking about 4 subphylums. So we're in the superphylum Ecthesizoa, phylum Arthropoda, and then from there we have 4 separate subphyla that we're going to talk about. So I mentioned that this is a hugely diverse group of organisms. So we have roughly almost a million, I think it's like 1,200,000 species now that have been described. And many of those are in the subphylum Hexapoda, or as many of you know it as insects. the first subphylum is Chelicerata, and so these are horseshoe crabs, spiders, mites, ticks, and soldifuges. And so these are the most primitive arthropods. They have 6 pairs of cephalothoracic appendages. Many of you probably know spiders have 8 legs, so we consider those 4 pairs. They also have a pair of chelicerae and a pair of pedipalps, and so these are structures that are located right next to the mouth, and they aid in feeding, kind of like having a hand right next to your mouth that you just use to draw food in. These organisms don't have antennae. That's a development that we'll see later on. And then they eat liquid food from prey. So if you've ever seen a spider eating a insect that's caught in its web, it's not actually digesting it inside of its body. What it's doing is it's spitting out like an acidic fluid onto that insect, and then it's sucking away that fluid, so those dissolved insect bits. So they're a neat group, fairly diverse, extremely diverse when we start talking about the arachnids. So there's 2 classes. There's Arachnida, which I just mentioned, and then the class Meristomata. And so Meristomata contains 2 separate groups. One's extinct and one's extant, meaning still present in today's world. So the extant group is, or are, the horseshoe crabs. And so this is what you would see. They're very common where I'm from. So every summer, generally in late June or early May, late May, early June, they climb up onto the beaches in order to mate and lay eggs. So the extinct group is called Eurypterids, and so they look very similar to Meristomata, or to horseshoe crabs, but they're extinct. And so they used to be marine organisms with these side appendages, but all the organisms in this class have a huge outer shell, or shield as it's called, that covers the cephalothorax. And so that cephalothorax is a combination of the thorax and the head. So they have this shield here, the cephalothorax, that covers the cephalothorax, and they also have what's called a telson. And so this spike at the end, right here, is what we call a telson, and so that's used in these horseshoe crabs to flip them over if they are upside down in the surf. So when they're coming up onto a beach to mate and lay eggs, they are trying to move against the surf, so as waves are constantly crashing over them, they get flipped upside down, and they can use this telson to flip them right side up. So the other class is the class Arachnida. This is an extremely diverse class. I think there's roughly 110,000 some described species, and it's probably much more diverse than that, just in terms of mites. Mite diversity is extremely high. We have mites on our bodies right now. I forget what genus it is, but they inhabit the pores on our face or on our body. And so all the organisms in this class have 8 legs. They have those 4 pairs of walking legs. But unlike class Meristomata, the pedipalps and the chelicera are modified. So the pedipalps are modified to bring food closer to this organism. So these 2 arms on this peacock spider are modified to bring food closer to the mouth. And then the chelicera are modified into almost like fangs, and they're used to inject venom into any prey items of this organism. So the class Meristomata generally depends upon external fertilization. So when they're climbing up onto the beach while they're still in the ocean, they're releasing either sperm or eggs, and they're waiting on how would you say it? Yeah, I don't know where I was going with that. The class Arachnida, reproduces mainly through internal fertilization. And so if you've ever seen David Attenborough's, I believe his invertebrate show, I forget what it's called. Mine skipping thinking straight. Anyway, these peacock spiders and plenty of other members of the class Arachnida have very complex courtship rituals in which they try to attract mates. Class Meristomata doesn't have this. So horseshoe crabs, so in the class Meristomata, are surprisingly fairly critical for making vaccines. So there's a chemical compound called limulus amoebocyte lysate, or LAL, and it's used to detect the presence of endotoxin or bacterial contaminants. And so when this the blood of this horseshoe crab encounters that endotoxin, it clots around that contaminant to form a solid mass. And so these horseshoe crabs are the only known natural source of this compound. So pharmaceutical companies collect them and then they drain their blood. And so this is an issue for the conservation of these horseshoe crabs, because when they drain their blood, they either do this where they're dead and all the blood flows out into these containers, or they draw the blood from them while they're still alive, and then they release them back in the ocean, but they die there soon thereafter. So this is a conservation issue for these horseshoe crabs, and their populations are declining fairly steadily since this was discovered in 1956. So who knew that a group of arthropods would be key to making a vaccine, actually, including the vaccine for COVID-19 too. So there are a couple alternatives, but in the US they haven't been proven safe for medical use. So we still in the US rely on LAL from horseshoe crabs to make sure our vaccines aren't contaminated. All right, so this is the subphylum Arthropoda. So this has two separate classes, the class Diplopoda and the class Chilopoda, and these are the millipedes and centipedes. So they have a fairly simple body plan. They have a head, which is at the very front of their body, and then they have a trunk. And so that trunk is the length of the body just posterior to the head. They have several pairs of legs. So in the class Diplopoda, they have 2 pairs of legs per segment. They don't really have, 1,000 legs per individual, as a millipede or the name suggests. They only have 2 pairs of legs per segment, which gives it this appearance that they have thousands of legs. And the class Chilopoda, or the centipedes, they actually do have, fairly close to 100 legs. I think it ranges from like 25 to 300 legs, but the main distinguishing factor is that they only have 1 pair of legs per segment. They all have these mandibles that they use to find prey if they are predaceous. So the majority of these organisms are herbivorous, so they're eating plants, but on the rare occasion there's that exception where they do have a predaceous centipede. So the main difference that we have of this group and all the other groups is that this only has 1 pair of antennae. So as you'll notice, as we go through these subphyla, one of the key distinguishing factors is the number of antennae. So the subphylum Chelicerata that we just saw in the previous couple slides, they don't have any antennae. This subphylum has 1 pair of antennae, and then the next subphylum that we talk about, the crustaceans, will have 2 pairs of antennae, and then the insects will have 1 pair or the hexapods will have 1 pair of antennae. So I mentioned that the majority of these diplopods, or not diplopods, these myriapods, are herbivorous, but there's an exception. There's always an exception to most things in biology, And so here's a predaceous cylopod or centipede that was attempted to be eaten by a pygmy rattlesnake. And so this pygmy rattlesnake and this centipede obviously fought quite the battle to the death. So you can see this is what, 16 inches? That's an extremely massive centipede. I don't think I've ever seen anything quite that large. And so this is found in the southwest. And you can see those mandibles that I mentioned on the other slide right here and here. And so those are fairly modified structures used to grip onto prey. You can see that they are extremely sharp. So that's probably what it used to tear itself out of this snake. So I forget which where I got this photo from, but it's not mine. Yeah, I forget where I got it from, but it's a neat photo and it's a pretty neat backstory too as well. So on to our third subphylum. So this is the subphylum Crustacea, and so this is like the crabs, lobsters, shrimp, and crayfish. Those aquatic crustaceans. It also includes terrestrial ones such as isopods, amphipods, and the like there, and some crayfish too. So they have paramandibles, kind of like what you just saw on the previous slide with that centipede, but they have the most distinguishing characteristic here is that they have two pairs of antennae. So you can somewhat see it in this crayfish here at the bottom. He has one pair of antennae here, and then he has his second pair going vertically right there. So this is an extremely diverse group. I forget what the total number is. I think it's like 60-some thousand described species. And then most of them are aquatic. So these guys all have gills, or book gills as they're called. And so those gills need to be moist or surrounded in water in order to be absorbing oxygen and diffusing oxygen out of the body, and then passing that oxygen to the rest of the circulatory system. So that's why these organisms are mostly aquatic, and that's why you really only find these isopods or these roly-polies in fairly moist soil. So another interesting group here at the very bottom, we have the mantis shrimp. And so most of you have probably heard about this, is that they have they have the most complex eye in the known natural world. So in our eyes, we have 3 cones, and we can see, well, basically the visible spectrum of light. These mantis shrimp have 16 cones, and they can see polarized light, UV light, as well as the visible spectrum. So they have an extreme amount of sensory input that's coming through those eyes to that nervous system. So that's a neat group. They have a couple different classifications as well. So they have, within these mantis shrimp, they have ones that will like stab their prey. They have ones that will punch their prey, and there's some other classification that I forget, but mantis shrimp are pretty cool. You can't keep them in aquarium unless you have these really special glass walls, because they will punch themselves out of a aquarium if they see a reflection. So they are a neat group. This brings us to the subphylum Hexapoda, and I think I'm going to spend probably the next 20 slides on this group, because this is the largest and most diverse class in Arthropoda, or I'm sorry, in most diverse subphylum in the phylum Arthropoda. So these organisms only have a single pair of antennae, which you can see here. And so that antennae is used as like a chemosentry structure, and so it's picking up pheromones or hormones left behind by other insects. So it acts as a communication structure as well as a way for them to feel around their environment. They also have a pair of mandibles, And so you can see this distinctly in this crazy ant, which is an invasive species in North America. So you can see these mandibles that it uses to catch other prey. These guys have 3 pairs of legs. They're all located on the thorax. And then they have 3 distinct body regions. So they have a head, thorax, and an abdomen. Whereas in the past class, Malacostraca in the phylum or subphylum Crustacea, they had a cephalothorax and a abdomen. So, like I mentioned, this is the largest class in Arthropoda, the largest class in Hexapoda too. And so insects cover 900,000 species, roughly about half of which is solely beetles. So there's a neat quote from J.B.S. Haldane, who's another evolutionary biologist with an extremely neat backstory to him. But when he was asked what science has described about a creator, he said that that the creator would have a special affinity, or I forget how he phrased it, an inordinate fondness for beetles and stars. So two most numerous things on this planet are beetles and stars, according to him at least. So roughly half the known species are beetles, and most of those are extremely similar in how they look. So if we look at the relationships within the subphylum Hexapoda, there's a bunch of subgroups. So there's 2 actual classes within Hexapoda, 2 major classes that is. There's Entogonatha, and then there's the class that we're all familiar with, Insecta. So the class Entogonatha, which is one that you don't need to remember, this is, or these organisms have internal mouth parts. This class includes things like springtails in the order Columbella, proturans, and Dipleura, so very primitive hexapods. The class Insecta has external mouth parts, so they have those mandibles, which is they use to catch prey. So I mentioned beetles right here. This is the most diverse group that we know of so far, and so they're in the order Coleoptera. Though there are some reports coming out that papers coming out that parasitic wasps, so a subsection of the group Hymenoptera, which is right here, there's some reports that those might actually be the most diverse group, which would surpass the beetles by a long shot. But as you can see, just in the sheer number of orders that we see today, there's tons. So there's everything from primitive insects that are wingless to winged insects. There's the very primitive winged insects like the mayflies and dragonflies, which have 2 sets of wings that are held rigid, that are non-folding. Then we have folding wings, 2 pairs of folding wings, and then a single pair of folding wings as well. So tons of diversity. Before I get into diversity or taxonomy, we'll talk about a little bit about anatomy or function of organs. So the reason why you don't see ants as big as a skyscraper is because they're limited by that respiratory tract. So I mentioned during our osmosis lab about the surface area to volume ratio that really is controlling the size of life. And so this is a really good example. These insects don't have an active way to transport oxygen or carbon dioxide out or into their body. They just rely on diffusion of gas that's flowing rather effortlessly through our called trachea. And so these trachea are small tubes with an external pore that run down the length of their body. And so from those trachea, gas is flowing in terms of oxygen, it's flowing in, and it's getting directly deposited to the muscles where it's needed. So back in the Carboniferous period when, or not Carboniferous, yeah, Carboniferous period, when oxygen levels were extremely high, we had Megacellura, which was that dragonfly-looking insect that had about a 3-foot wingspan. So those high oxygen levels allowed the insects to grow to fairly extreme sizes, well, compared to today's insects. So as the amount of oxygen in the atmosphere has dropped, insects have remained small. So if your irrational fear of extremely large, or if you have an irrational fear of extremely large insects, you really have nothing to worry about. So if we go into the excretory system is dependent upon what are called Malpighian tubules. And so these tubules run throughout the body, collecting metabolic waste. And so this waste is then transported to the front of the gut, where it's then mixed with food products. And so nutrients and water are reabsorbed, and then the true waste in the form of uric acid is excreted out the rectum and anus. So that's the excretory system in hexapods. So I mentioned that there's roughly about 1,200 some known, or I'm sorry, 1,200,000 some thousand known described species. So this is an extremely successful group. One of the reasons why this group has been so well documented is mainly because this is a fairly charismatic group. So science likes to deal with organisms that are fairly macro, so something you can see with your naked eye. Also, beetle collecting was a fairly popular sport in the, well, not really sport, but a hobby in the early 1800s. So Charles Darwin was a very avid collector of beetles, and even aristocrats in the 18th century or 19th century were collecting beetles as a way to show that they were well traveled. So just in terms of why we know so many of these insects is because they're fairly well studied through history, just through having charismatic or scientifically charismatic appearance. So plus beetles are extremely colorful and cool. Most people consider them very pretty. So they're the jewel beetles that have very ornate shells. Also, many of these insects act as agricultural pests, so there's become a lot of research into different pests such as the aphids, which will eat roses and stuff, will tear up your garden, and all the way up to butterflies that will, as a caterpillar stage, will destroy whole orchards. So there's been a lot of research on this group. But even still, they're just an extremely successful group compared to any of the other subphyla or classes or phylum that we've talked about before. So there's a couple different reasons for this. The first reason is that they have those Hox genes, which are a subset of homeotic genes. And so these Hox genes are regulatory genes that are coding for fairly distinct regions of the body. And so if you look at this chart on the right side of the screen, we can see that there's Hox genes that are coding for each one of these segments. And then throughout time, as these organisms evolve, Hox genes were condensing into the anterior region of the body, so towards the head. And so as these Hox genes are condensing towards the head, they're expressing and controlling where certain structures are going to be placed. And so as these Hox genes are condensing into this region of the head, it allows for a very wide variety of mouth parts. And so these organisms, or these insects, just by chance, have had the ability to adapt to almost every single food source that's on this earth. So that's responsible for Hox genes and how they condensed over time through tagmosis into the head. And so there's a couple neat studies using these Hox genes and fruit flies. So the most popular one, or that you probably heard about, is just Drosophila melanogaster, or the common fruit fly. And so they can alter these Hox genes for each one of these segments, so either the head, thorax, or abdomen. And so they can alter the Hox genes that are used for a antennae, and they can show a leg there instead. Hox genes are a really cool way of showing evolutionary development. There was a study, I forget when it was, 2015, something like that. Either way, this guy was studying mutations in chicken embryos, and so just through a single mutation in one of these Hox genes, the chicken embryos started to grow teeth, just like they would if they were a dinosaur embryo. So you can see these Hox genes controlling for certain regions. When they get turned on or off, it can have pretty substantial impacts. So that's a little bit of a side note, not really related to insects at all, but well, slightly. When these insects, or when these Hox genes mutate, or there's a mutation that affects these Hox genes and how they're regulated, you can have fairly diverse changes in how different parts of the body are expressed. So Hox genes are probably the number one most important reason for why these insects are as diverse as they are. All right, so as insects became more complex with increasing diversity, or over evolutionary time, one thing that was changing was the actual positions of their wings. So in the most basic insects, we have organisms or insects that lack wings. So this includes such organisms like the Archaeognatha or the rock crawlers. So you can often see these organisms, I believe it's on the next slide too. You can often see these organisms in areas where there's lots of shale or exposed rock, along streams or along what is it called? Gravel beds of streams. So the most basic organism or insects lack wings. If we move on to somewhat more derived organism or insects, they have two pairs of rigid wings. So if you look at a mayfly, they'll have wings that are held vertically, so straight up and down. And so those are very closely related to the dragonflies, which are shown here and here, which have their wings held directly out horizontally. So these two pairs of wings that are held rigidly are not folding, so they're just held out to the side. So I just mentioned mayflies, but another insect, such as these damselflies here, show that that pair of rigid wings being held vertically, or at least at a 45-degree angle. One of the groups the group that I study, stoneflies, has this characteristic. So they have two pairs of wings that are folding. So they take that wing, they have an extra set of veins that will either pump full of blood or restrict blood, and so they will fold in on themselves, protecting them as well as giving them a much more streamlined body. The most derived characteristic is having a single pair of wings that folds. So if you look at a bee or a fly, they'll only have one pair of wings that will fold over the abdomen. They'll also have what's called a haltere, and so that's a very modified wing that's used for stabilization during flight. So one of the reasons why insects are so successful, especially the more derived groups such as the beetles and the parasitic wasps or Hymenoptera and bees, is that because they can fly, they can travel long distances, they can catch other prey items out of the air. So wings are another reason why this group has been so successful over time. So I mentioned Archaeognatha. These are the rock crawlers. This is an extremely neat group of insects. So these insects have scales. They don't have just a smooth surface like all the other insects that we'll see. So this order is Microcophrylia. These are rock crawlers, really neat group of organisms, and these are the most basic basal group of insects. So just to show more of the wings, you have mayflies, which I mentioned here. They have the wings that are held straight up and down or vertically. You have dragonflies, where those wings are held out to the side. And both of these are not able to fold their wings, so these are held rigidly. Here we have a beetle, and so it has this single pair of wings here that will fold over its abdomen, and it has a modified pair of wings, which we call elytra, that will cover those wings, keep them dry, and protect them. Here in this crane fly, we have a single pair of wings as well as a pair of halteres, and so those halteres are aiding and stabilizing this organism while it's flying, and it can also fold these wings. So these two are the most derived, most recently evolved, and these two are much more basal organisms. So moving on from why they're successful, we'll go through the smallest known insects, the largest known insects. So the smallest known insects that we have found so far are wasps in the family Mimiridae, and so these are known as the fairy wasps. So these are found in tropical areas. I believe Tinkerbella ninata was found in I believe Puerto Rico, and then this one over here was found in Hawaii. So these are extremely small organisms. I accidentally cropped out the scale bar here, but it's something along the lines of a fraction of a millimeter across. So I like to say that physics gets pretty funky when you're extremely small. So when these guys are flying, they don't have these long lobed wings that you see in other insects. Instead, they have these rows of setae or hair that it's using to literally climb through the air. So as you get fairly small, air becomes much more dense relative to your body. So instead of having to just create substantial amounts of lift, they have to pull themselves along like they're crawling through honey or molasses. So they're fairly neat organisms. Don't ask me how they caught them. I don't know, because they are extremely small, much smaller than a normal net would allow for catching. So that's another neat group of insects. The largest known groups of insects, if we consider beetles, this is Megasoma acidion. If you consider butterflies, this is Ornithoptera Alexandria, or the Queen Alexandra's birdwing. So this is found in Australia, and I believe this one, the Megasoma, is found in, I believe, Brazil. So fairly tropical areas. I believe this one, Ornithoptera Alexandria, is on the brink of extinction. One, it's a collector's item, and two, it is just so large that it's not flying very far. So it's subject to predation from birds as well as loss of habitat. So two more neat groups as well. So another reason why these organisms are so successful is many of them have developed some way of camouflaging themselves through Crypsis. So if you look at this example here, if you try and find the butterfly, you have to give it a little bit of a look, but it's right here in the center of the screen. So this is Kilima inachus, and so its body resembles, when the wings are closed at least, its body resembles a dead leaf. So as a predator is investigating this dead leaf. If it continues to investigate and isn't really put off by a dead leaf, this butterfly, Kallima inachus, will flash these bright colors, so these look like eyes, in hopes of deterring that predator once again. So Crypsis is a neat way of camouflaging organisms. So one of the more popular examples of this is a stick insect. So these are native to Kentucky. They look just like a branch. They walk extremely slow, and they mimic the movement of a branch moving in the wind. So as they stay still, they don't just stay still on a branch. They start swaying back and forth to mimic how a branch naturally moves. So I believe this is the last one that I put in this slideshow, but you can definitely tell that insects are extremely successful for a variety of reasons. And so this reason is probably one of the more important reasons. And so insects have had a close association with plants, especially flowering plants. So flowering plants evolved roughly 120 million years ago, and ever since then, both plants and insects have had really a symbiotic relationship. So if you think about it, organisms like a honeybee or a carpenter bee have a very broad range of flowers that they are taking pollen and nectar from, but there's many more species that are specific to a single plant. So if you look at these leaf hoppers from Hawaii, each one of these leaf hoppers are a different species, and so these different species are unique to a single plant and to a single island. So if you throw in a little bit of island biogeography into here, once you get away from that mainland island of Hawaii, forget what island that is, you get less and less species. But on each of those islands, you have unique communities of flowering plants, and each one of those flowering plants has a specific leafhopper that is feeding on it or taking advantage of some aspect of it. So this host plant specificity has resulted in radiation of a diversity of insects. So there's a couple more things I'll talk about. One of the things is metamorphosis, so the changing of insects from, or even crustaceans, from the larvae to an adult, and from the eggs to a larvae. So the first one I'll talk about is hemimetabolous. So hemimetabolous individuals have larvae that look identical to the adults. So if you look down here, we have a true bug in the order Hemiptera, and so that Hemiptera is not changing very much as it moves from each one of these larval stages to an adult. And so if you look at the adult and nymphs, one way you can tell the nymphs apart is that these adults have functional wings and these nymphs do not. So if you look at a dragonfly, dragonflies larvae and dragonfly adults look fairly similar. The difference being is that these larvae only have wing pads. They don't have true functioning wings like these adults do. So if you look at this picture, you can see the trachea, these tubules, those thin white lines that are being pulled out of the adult dragonfly as it's emerging from that shedded exoskeleton. So that exoskeleton is going through, going into the respiratory tract, lining the respiratory tract, and so even that is shedded or shed from the body during ecthesis or metamorphosis. So another type of metamorphosis is holometabolous. This is probably what you're more used to. And so if you think about a monarch butterfly, it starts off as an egg and then moves to a larval stage, kind of like a caterpillar. From there, it goes to a pupa stage where it's inside this chrysalis, and so tissues just kind of dissolve into this goo and then become rearranged. And given some time, after everything's become rearranged, that butterfly, or that caterpillar will emerge from that chrysalis as a butterfly, or as an adult. So next week is the midterm. I know there's a lot to cover in this this video, but like I said, next week's the midterm. There will be 2 study review sessions. There will be a review session on Friday and a review session on Tuesday, the day before the first midterm. So Friday of this week, I'll send out an email sometime Monday or Tuesday with the times for Friday and of the review sessions. So the way I envision you guys taking this midterm is that there's a 2-hour block of time where you're supposed to be in this class. Whichever hour you're not actually in here taking the lab practical, you'll be taking the written portion online using Blackboard. So hopefully you guys will all be using the computer labs or bringing your own computer and taking it on there. So that's how I envision it happening, but we still have a week and a half before we have to deal with that. And by then I'll have a better idea of what to plan for.